I'm feeling very happy to participate in this. Giving a lecture in here. Um, uh, I thank Dr. Jenny and Varghese for organizing and putting up a huge number of organizer and making such a wonderful conference, wonderful lecture series. And I thank Dr. Sibu Simon for inviting me to this lecture series. Um, I'm going to talk about a computational approach on epigenome editing by using a genomic or genetic scissor, what do you call it as a genetic scissor, which is a CRISPR-Cas9 system. um, this presentation have little bit of a, it's not about directly about CRISPR-Cas9 system. It's about epigenome editing. Okay, genome editing is another area by CRISPR. Epigenome editing is another area. Okay, and so the first complexity is CRISPR. Maybe for some of you it may be new, and the second is it's about epigenome editing. The second complexity, and the third one is uh, this is just an introduction, but I'm going to talk about CRISPR broad. That's a kind of a novel tool, or what do you call it, a bioinformatics software that we have designed. Okay, so it will be three-way complex. I only can assume that. Also, it's about computational approach. So I will not have any, gel or images, something that is very friendly to your eyes. Uh, so just try to stick on to me. I will try. I will, some of the slides may be a little complex, so you can avoid and just grab a few ideas that we are trying to put in. Um, so for the newcomers or somebody who want to take home a simple message, what I'm trying to do is the DCAS9 is a CRISPR DCAS9 is a system that targets a particular region in a genome. Which is here is the ladder, which is nothing but DNA sequences. Okay, if you call that as a DNA sequence, and the scissor is the CRISPR-Cas9 system. Okay, that's called a molecular scissor. Uh, Everybody knows that restriction endonucleases are molecular scissors. So the DCAS9 is nothing but a restriction endonuclease. So this molecular scissor can be used to, for example, in a condition, for example, in a healthy human, it's like this, the top one. But in a diseased condition, you may have some kind of deletion, okay, some kind of deletion in the nucleotide, some kind of insertion in the nucleotide, or some kind of modification in the nucleotide. You all know that is nothing but a mutation. Okay, so this mutation can be rectified by using this molecular scissor. So the whole idea is the disease can be the mutation can be this three ways, like deletion, insertion, or modification. So these three ways can be you know uh, rectified by using this molecular scissor, which is nothing but CRISPR-Cas9 system. So that's the whole idea. Okay, this is a genetic scissor that can be utilized to ward off few diseases. For example, cancer, people use it for a cancer. There is no human application so far. Okay, there is no direct human application. It's mostly on a certain model system. For example, a plant system, which can you can use it easily. In human system, it's very difficult to directly use the CRISPR-Cas9 system, but people are still trying to use it on cancer and other stuff. Um, so that's the nutshell. Okay. uh, okay, and I put my presentation this way. Okay, it's like an introduction to a little bit on CRISPR, and then a little bit on what are the technologies that CRISPR can be applied, and then I'm going to go a little complex on epigenomic editing, and And what we are doing is particularly the guide RNA design. Okay, guide RNA. I will talk about the guide RNA. What is guide RNA? What is its role in the DCAS9 system targeting? And then finally, maybe I'm going to more emphasize on the CRISPR broad. That is a guide RNA design tool that we have designed. I have written the program, so I will talk more on that CRISPR broad. Okay. um, each of them have a subset. Okay, I mean subtopics. Those are like introduction to CRISPR9 system is about what are the components in the dcas9 system, what are PAM sequence, what is guide RNA, what is dead cas9. So these are like component of the dcas9 system. And then the applications of CRISPR includes genome editing. Genome editing is nothing but, right now before slide I have spoken about it. It's about just changing the nucleotide. And then there are other applications too, live imaging of DNA, transcriptional control of the genome, and so and so. And then in the epigenome editing, I'm going to talk about um, uh, what are the kinds of epigenome modification that can happen in a genome, how this can be modified, okay, or retrieved back. And then the guide with the methods of guide RNA design. This is mostly computational method. So I'm going to talk about what are the features or what are the properties of that guide RNA that can be utilized to make it a better guide RNA or an efficient guide RNA. So, and then finally, I'm going to talk about the tool that I said. So in that, what I'm going to talk about is what is the strategy, what is the modules that we have adapted, and how we have applied it to what you call C. elegans, which is nothing but a worm. So we took two species and then tested our hypothesis on those two genomes. And then I'm going to end up saying that this tool is much better than I mean our tool is the best, so something like that. So that is like highly selectable guide RNA. So this is my flow. You may miss few slides, but just to try to go along with me. um, okay, the eukaryotic cell is completely controlled by the genome. Okay, the genome is the main player that is like the controlling the central dogma of the cell. For example, here it's in the nucleus, and then sitting inside the nucleus, it controls the transport of the proteins and others, and also it controls the transcription, translation, and also post-translational modifications. All these things happen under the control of a genome. Okay, genome, which is nothing but um, a sequential arrangement of nucleotides. Okay, so anything which is like anything that happens to this sequential nucleotide arrangements, which is nothing but primary sequence of a genome, can heavily alter the function of the whole species. Okay, this is implicated in both health and other diseases. So these are nothing but these nucleotide changes, that these are Mendelian inheritance. That means it's like
Sir, you are muted actually. Hello? Hello?
okay. I think I'm audible to you now.
Yes, sir.
Yes, sir.
Yeah.
Yes, good, good, good. Yeah.
Yeah. Yeah, maybe the when change the slide to one slide, maybe it got muted. So, uh, okay. So any changes in the nucleotide can heavily affect the function of the cell. So this is nothing but a Mendelian inheritance. I'm going to talk about one more one more changes to the genome that can also affect the function of the cell. Okay. Okay, that is non-Mendelian inheritance, which is nothing but apart from the genomic nucleotide changes, there are some few other groups or chemical groups that can be added to the already existing nucleotide. The nucleotide don't change. For example, the ATGC remains the same, but to the A, T, and G, and C, to these nucleotides, there will be some chemical groups added, and then that controls the transcription of a cell, okay, of a genome. Let's see what are those. Take for example, this is your chromosome. The chromosome is wound like this, and this is for example, if you take the primary sequence, that is nothing but the DNA. This DNA sequence coiled together through the histone proteins, which is nothing but just an octamer of proteins, and then that coils again to tertiary structure, what you call it as a chromosome. Okay, so the primary structure is the sequence of the nucleotide sequence. The secondary structure is nothing but a looping, and the tertiary structure is nothing but a chromosome. So this within each of this structure, a chemical group can be externally added. For example, this is like a GC. The nucleotide doesn't change. There is no mutation here, but there is a methyl group added to the nucleotide. This is which is nothing but an epigenetic modification. Genetic modification is mutation, and the epigenetic modification is adding additional chemical group to the existing nucleotide. And then apart from the DNA a modification on the DNA, there could be modifications on the histone proteins too. For example, these are like a wound around this octamer protein. So these modifications on the histone proteins, for example, I put the figure shows there are a list of chemical groups that can be added. Okay, acetylation, phosphorylate group, sulfur group. Any these groups can also alter the protein function, which is wound around that DNA. So this way, again, this protein have chemical group added, but the nucleotide remain the same. So uh, both the genetic modification and also epigenetic modification can alter the function of the cell. So how this is also dynamic. Okay, this is like a very dynamic. Okay, this modification, for example, the mutation is very localized. Okay, in one nucleotide, in a particular sequence, there will be changes. But for the epigenetic changes, for example, this is a whole gene. Okay, 5' to 3' end. In the whole gene, the whole these are list of histones that is wound around the DNA in the whole genic regions. So all of the histones are modified with these modifications. For example, these are like a chemical group added to those histones. So this is like a very longer. For example, the average gene length is, for example, 2 kb to 3 kb. Okay, 3 kb is the average length. 3 kb can have 3 kb mutations. Okay, 3,000 mutations. So that many number of mutations is possible, but here the modification is like through the gene. So you cannot target such a region like what you see. CRISPR can be directly applied to the mutation, but it's very large here. This large region modification, this is very difficult with the CRISPR. So there needs a novel strategy to target this kind of large modifications. Um, Also, as I said in the earlier slide, that these are like histone proteins, and this is the red one is the DNA. So this DNA, these modifications, in case of mutation, it's irreversible. But in the epigenomic modifications, if you add a chemical group, and that chemical group can also be erased over time, like in a specific for example, in children there are some modifications which is happening, but in adults those modification will be erased out. So this is like a dynamic process. So certain kinds of genes are expressed in one condition, and in another condition, the certain number of genes can be expressed. So this is like a dynamic process. So this is like a goes forth and back. So that's in a cell, you have certain enzymes that modify certain histones that will repress that gene. Okay, the gene have the histones wound around that. So the modifications based on the modification on that gene, it can either have an expression, or it can have a repression. Okay, it can be activated or repressed. So these are, for example, the oranges are the list of enzymes that can modify that histones underlying that gene that can lead to repression. So these are the list of enzymes, the green, the compass, dot, BAPON. These are like enzymes that can modify certain genes that can lead to activation. So in a cell, there is a fine balance between activation and repression in a particular state. For example, if you take a young one and an adult, there will be a certain level of changes that will be harmonized for on that stage, for that stage. So I just want to summarize here that there will be there are one part that is like genetic changes, that is like changes in the nucleotide. For the case of epigenetic changes, there is additional groups that is added to that. Both can lead to a similar activation or repression. Okay, so what these kind of changes can be eliminated by using what we naturally. For example, the mutation is like if you smoke, or if you have some kind of disease, then you have this mutation. So you use CRISPR-Cas9 system to eliminate this mutation. For example, here the G have changed to T, so you can put back the G here to under and then revoke the disease process. So in the same way, you can use certain enzymes like I saw in the previous slide. You can use certain enzymes that can that can remove certain modification and add another modification. So there are certain enzymes that can remove this methyl group from this nucleotide, and thereby you can reverse the disease. So these are the two aspects that one can use CRISPR-Cas9 system. So okay, let's see what is the CRISPR-Cas9 system. This is a CRISPR, diagrammatically represented, but actually it's a protein and a RNA component. Okay, it's a complex of protein and RNA. Okay, just as simple as that. So Cas9, okay, let's see CRISPR. What is CRISPR? CRISPR is nothing but is based on a sequence. Okay, it's nothing but DNA sequences which are very short and repeated in a genome. Okay, uh, so what we are trying to do is we are trying to use this Cas9. Cas is nothing but CRISPR associated protein 9. Okay, that's nothing but a protein name. Cas9 is a protein name. So they're using that CRISPR DNA sequence along with this Cas9 protein, okay, to target a particular region. Okay, to target a particular region. What is targeting? Is how we can target a particular region. Well, because the DNA is too long, like 3 billion bases are there. How we can target a particular region? So you use something called as a guide RNA. Okay, the guide RNA, nothing but here. So the black one is a guide RNA that will take your Cas9, the Cas9, CRISPR-Cas9 system to a particular region in the genome. So this guide RNA is complementary to it's like a sticky. Okay, it's like a stick. So you say that you give it's like a courier system. So to the courier box, you just give the address, right? So the same way to this DCAS9 box, you write an address, like the address is nothing but the guide RNA. So it will directly match to the target and then edit it. So when you know that you have the mutation in the earlier slide, when you know you have a mutation in T here, so you specifically design a guide RNA that with this sequence, like maybe you can select TCC, TA, and then attach to the Cas9. Okay, this is nothing but an guide RNA, and that nucleotide you attach it to the Cas9 system, and the Cas9 go and may modify that. Okay, so that's a kind of tag or an address system, a kind of barcode that one use that is the guide RNA that one could use to target a particular sequence. So there is a problem here. Okay, the guide RNA, people cannot use a guide RNA which is like because all the genome sequence is kind of repeated in multiple times, like there are a lot of repeat sequences, because the whole genome is compiled of what, four nucleotides in a different combination, but you will find that kind of sequence throughout the genome. Okay, but if you want to edit a particular nucleotide, you have to target these sequences using this guide RNA. You cannot just randomly put a sequence. It will because there will be off target. Off target means you want to off target means you want to edit this T, but it goes edit the first T and the last T. So you should avoid that. So you need a kind of complementarity and then a kind of a specificity for the guide RNA. So that's important. Okay. The protein can go anywhere. Protein, the Cas9 is nothing but an endonuclease. It can go and cut any T, any nucleotide if it goes and bind. But you need to have the specificity using a guide RNA. Okay, that's the important thing. So this guide RNA, people, it can, and also the guide RNA cannot be very long. Okay, it cannot be very long. Then it cannot take the DCAS9 to the, for example, just like addresses. If you write address like a 10 address at a time, you cannot deliver it, right? It's the same thing here. So just a unique target guide RNA is needed. So I just want to give an additional more information. So that just the DCAS9 component. But this component has been described first in bacteria. Okay, first in bacteria, they isolated this system from a bacteria and they randomly used it on prokaryotic sequences, and finally they find that this could be applied to eukaryotic mammalian system too. And then currently it can be, it's still in progress, but it's applied to human embryos currently. Hopefully it can, down the line, you can see a lot of applications for CRISPR for multiple diseases. Also, these two ladies, Jennifer Doudna and the other person, they got the Nobel Prize in 2020 for that, discovering this epigenome, the genomic editing tool. Also, this editing tool is it's very simple. Like they initially find only that DCAS9 and a simple 20 nucleotide guide RNA based arrangement. The whole system is like that. But there are n number of tools currently that can take a little larger guide RNA, and also that sometimes the specificity is like very too narrow, or sometimes the specificity is very low. So you can modify, instead of DCAS9, you can use other Cas enzymes. There are many other Cas enzymes. There is a family of Cas enzymes. I'm not going to cover that here, but there is a huge number of enzymes people are using. You can see from the number of plasmids people are ordering that it's getting higher and higher, and there are a lot of a family of Cas9 molecules being discovered. So there's a huge potential for this application in the future. And I'm going to show one slide on how this can be applied to in a particular sequences. Okay, take the case here. The brown one, if you take the top one in the figure, the brown or the gray one is nothing but the DCAS9 enzyme, which is a protein which is very huge. Okay, it's a huge restriction endonuclease. And this pink one inside is the guide RNA. Guide RNA is nothing but the which should be complement to the targeting sequence. Okay, that is the guide RNA. That is around 20 nucleotide in sequences, and if you see, they have a specific spacer which is called nothing but NGG. uh, so these are like apart from the complementary to the 20 nucleotide being complementary to the target, it should also have this feature, which is nothing but PAM. PAM is nothing but a kind of a protospacer associated motif. Okay, this is like NGG is nothing but a motif, nucleotide motif that should be conserved. These are like conditions for this Cas9 to go and bind exact for a specific binding. Okay, so the nucleotide should have, and what you see again, you see a kind of hairpin bend, right? Hairpin bend which is complementary to itself. So this is a kind of a complementary spacer regions. So these regions is nothing but a feature of this guide RNA. Okay, the dcas9 nuclease is just a functional nuclease. There's no modification to that, but you can use the same dcas9 to different targeting regions based on the features of the guide RNA. One is the complementarity, and the second one is the second structure of the guide RNA, which is nothing but the hairpin loop, and the third one is the PAM motif. So this NGG motif. So if you find these three feature, then the specificity of this guide RNA is a lot. Okay. Also, there is another feature. For example, in this alignment, for example, this is a kind of a, complementarity. What percentage is complementarity to the sequence? That is also matters. Okay, if it is 100% and there is no after, that is means it can bind only here 100%, but not to other regions in the genome with the same sequence, because this sequence can be repeated in the genome multiple times, as I said earlier. So if it is unique, then that's a good feature of the guide RNA. So when it binds, when you have the complementarity, then it brings the dcas9 system to that nucleotide, to that genome sequence. The 5' 3' here mentioned here is nothing but the genomic sequence. So what it does is, this is its target. Okay, the arrow, black arrow is the target of that, and then it cuts a double strand break, which is DSB is nothing but double strand break. And then what it can do is, it can do a insertion, or it can do some kind of correction. For example, if the nucleotide is changed, it can correct that nucleotide, or it can completely delete certain nucleotides, like 10 nucleotides, 5 nucleotides. You can delete certain regions. So you can insert it, or correct it, or delete it using this kind of system. The one is the complementarity, and then the denuclease just make a double strand break, and then there will be joining again. Before joining, it determines whether it is insertion, deletion, or correction. So that's how the system works. It's very easy to follow up here. So that is like for mutation, for one process, like mutation, you can delete it, insert it. That for nucleotide based modification. But as I said earlier, you have a second type of modification, which is epigenomic modification. Here, what you do is, there is no need for double strand break, but instead of that, you need to you need to change the add some kind of chemical group. As I said, you don't need to cut the nucleotide, but you need to add some chemical to the chemical group to the nucleotide. So in this case, for example, as I said, that I put an orange and apple diagram to show that there could be one for repression, one can be used for activation. So those molecules can be here fused to the uh, fused to this dcas9 enzyme. Okay, this is inactivated enzyme, but that there is an effector group, nothing but you can add additional enzyme to the dcas9, and then the dcas9 do not cut the nucleotide, but this effector molecule that you add additional enzyme can put additional group to that genome, and then it can activate or repress. Okay, so it's as simple as that. So one is directly cutting the nucleotide and eliminating the bad things. And the second one is without eliminating the only the chemical group without cutting the nucleotide. So for that, you need to add additional enzyme to that. Okay, to the dcas9. Adding is nothing but you make another protein, two proteins, and then fuse it. Okay, you have two proteins. One is the restriction and the nucleus. Another one is your activator. Then you fuse this protein and then put the guide RNA again. Then it goes and activate whatever the region that you want to. Okay. So the uh, and then this is the whole component of the dcas9 how it works. But there is this addressing system. This is like a you cannot write like just there is a format, right? There is a format for address writing: the person name, street name, and then state, and then country, and then pin code, right? There is addressing system. So this is like a feature of the address. The same way the guide RNA have certain features. So I'm going to talk about the guide RNA feature because that is the main component. The dcas9 and the effector molecule, these are like another component, but the guide RNA is a very important component. So this is like a 20 nucleotide in length. Okay, the guide RNA is like, for example, it's like a post office, postal box. You can have a certain number of boxes, right, for address writing. So it has 20 nucleotide, and there is a certain kind of chemical nucleotide composition. For example, the GC content should be very high. Okay, instead of 80, in the 80 GC composition, the GC component should be very high. And then there should be a kind of, at the end of the nucleotide, this 20 nucleotide, there should be a kind of motif in GG. Okay, this should be added to the nucleotide. So this will enhance its function. Okay, function means it's enhance its binding to that particular target. And then complementarity. You can design like a few nucleotide uncomplement to the non-complement to the target. So, but if we have more kind of 100% complement, and then that will be very specific. So, but still you can adjust the nucleotide to target, because sometimes you cannot design a specific guide RNA, because guide RNA design have a multiple number of problems. So you cannot design particular nucleotide. So in that case, you can adjust little bit to this complementarity. But the thing is, higher it is, higher the complementarity percentage wise, higher its function. The third one is the secondary structure. It should have at least one, this kind of stem loop 1, stem loop 2, stem loop 3. So this kind of structure it should have. When you design, you also take into account this stuff. So our idea is bioinformatically, why I am talking about this, our idea is bioinformatically design this guide RNA such that, as I said earlier, you can do multiple other processes with dcas9 system. So the guide RNA component is our focus. Okay. Oh, okay. This guide RNA, there are like multiple other tools. Okay, there are like multiple other tools that one can design. There is some unique thing that we have designed in this strategy of guide RNA design. For example, I'm going to give three example of already existing method. One is there is guide RNA that can be designed. One guide RNA can bind to only one region. Okay, that is their whole idea. So that program is like, you take one region, there is one, they want to design only one target, the guide RNA which can bind to only one region. And there is other people also design the second tool, that is like there's one guide RNA that can bind it more than 10 regions. For example, if you want to correct mutation of 10 regions, they can do with one guide RNA. Okay, because making an experiment with multiple guide RNA will not work. So you need a low number of guide RNAs. Okay, that's the whole idea. So they have one guide RNA and one target. That's the easiest way. But in the genome, it's not like that, because there are multiple other copies, as I said earlier. And then the other idea is multiple guide RNA to multiple regions. You can design 10 guide RNA and it can bind to n number of regions, 10 regions, more than 10 regions. So that's called multi-targeter. What we have designed is we took one region in multiple kbs, okay, like a long regions. All these things that I mentioned earlier for cannot edit a larger region, okay. For example, I want to edit 10 kb region, okay. I want to edit very large regions. In that case, one guide RNA is not enough because one guide RNA can target only to a particular nucleotide, okay. So we designed something like a one guide RNA that can bind in multiple regions in the same, in a very large region. Okay, it can bind to multiple location, but in a very large regions. That's our idea. But it's very restricted to one region. But you'll not find other, off targets. The OT means off target. The first one do not have off target, but the second one may have off target because it can bind to multiple location. And the third one here I mentioned that if you have multiple guide RNA, it can bind to multiple off targets. But our case, we want to restrict it to one guide RNA binding to one location, but not in the other locations at all. So we designed something like this. And so that's our whole strategy. One guide RNA, but multiple location, but within the same region. So we have designed that. So these are the list of already available softwares, but none of them have the feature that can cover the whole genome. Okay, we can go up to more. The largest they can do is 50 kb. Okay, they can design more 50 kb max, but we have exceeded like a huge regions, which can cover the whole genome. So the rest of them, I'm going to talk about how this tool is working. Okay, this tool, how it is working. So we have designed this software such that it takes into the account that the feature of the guide RNAs, GC content and the length and other stuff, and it maps to the genome, and we have given a score. I'm not going to avoid how we calculate the score and all what it means, but finally what our aim is to just you know get into optimal number of guide RNAs. Okay, that's our whole idea. So it have around 8 modules. The program runs in 8 modules. It's open source. You can download it and put it in Linux box, and then it's based on Python. It have 8 modules, and as I said earlier, the whole idea is finding a multi-targeting but a single guide RNA. Okay, I'm going to avoid the details of the work. So directly going to the result. So we applied these two, one to the C. elegans and one to human genome, just to check whether the C. elegans is a worm, which is nothing but around 100 MB genome, which is like a Arabidopsis, it's like a plant, like a small genome. And the humans is 3 billion base pairs, so it's a very large. So we want to test whether how it works on a smaller genome and a larger genome. And does it give a very huge differences? Okay. Um, when we ran the genome with C. elegans, what this C. elegans have 5 chromosomes. The blue bars are nothing but a 5 chromosome and one sex chromosome, autosomes and sex chromosome. So what we find is certain regions are highly covered by the guide RNA. Certain regions could not be, but this is a property of the genome because they have a certain regions which are very high repeats. But overall, what we see is you could cover at least, this is very highly dense, but you still see the blue bar are still distributed throughout the chromosome. So that means that you can still select the genome editing on the whole genome. Ideally, one would want that the blue, the thick color blue distributed throughout the chromosome, but we couldn't find it. What we see is a kind of dispersed distribution, but still we are able to cover the most of the chromosomes. We didn't miss any of the chromosomes, so it's distributed throughout the chromosome. And if you see the human chromosome, this is like the red length says that the length of the chromosome. Okay, the dot, whatever the red dot is nothing but the guide RNA presence. There are, as I say earlier in like C. elegans, there are certain regions which are very high in guide RNA. There are certain regions because human genome is very highly repetitive, so it's very difficult to find a unique guide RNA regions. Okay, so that's a part of the genome. You cannot change the genome. The genome is static, so we need to design based on that. But we are still able to find, we can target that most of the regions. The red dot is distributed mostly. But for example, here 13, 14, 15, start of the chromosome, you're completely missing guide RNA. So certain regions we couldn't cover it. And also we see there is like there is like a huge number of sequences with off targets. There are like although we didn't find unique sequences, certain regions also have off targets. In both cases, C. elegans and also in humans, we see that the guide RNA have both target, but also sometimes it's binding to other regions too. So uh, but if you select the ones which is not covered in the thick blue, those are like the light blue, those are like with off target. But without off target, if you see the thick blue, green, so those are like a number of most of the sequence have at least some kind of off target. But still there are a lot to select from. Then we arrive, we give the score based on the GC content, the kind of alignment, the kind of secondary structure. I said all the properties we included to give a score, because if everything is high, then the score is high. Okay, when you have more kind of unique alignment, then the score is high. When you have very high GC content, the score is high. So we give like that. So what we see here, the score for all the guide RNA is distributed throughout. That means you may not you find a certain guide RNA which have good score, but there are other regions which do not have a good score. So that's how it is. But if you see the higher numbers, those are like very highly negatively correlated. But if you see here on the right end, very highly correlated, there is a selectable, good selectable amount. This is against certain program which cannot find guide RNA at all. Okay. And also if you see you could expect that when you see zero off target, then you see that there is a very high number of there is a very good score benchmarking that when you do not have a unnecessary hit, then your score is good. So that means our score is still valid, so the feature that we defined is still very good. So again we see here, when you have a multi-targeting effect, when you see increase the length of the window, and this is the number of hits. So when you increase the number of the size of the window, and when you increase the number of hits, it gets reduced. So if you make it slightly shorter nuclear guide RNA, then you can find that there is a huge number of like this here in the blue color. So there is a huge number. So we see that there is a selectable number of guide RNA based on the score. Higher the score, better the guide RNA. Again we see the same thing in the C. elegans and the humans. The on target effect is off target effect is still found. So you see certain number of guide RNAs unique, but there are the red ones are targeting guide RNAs, the black ones are off target guide RNAs. Those are like huge in numbers. So it's like a guide RNA finding is a kind of finding a needle in a kind of huge maze. So, but still it's achievable. Still there is a, the red ones are achievable. So these are like kind of a guide RNA. For example, these are IDs. Okay, the numbers are nothing but the guide RNAs. There are more guide RNA in this region. This is like a chromosomal region. Okay, when I say CHR1, that means a chromosomal region. Okay, and the ID numbers are nothing but guide RNA. So in one region you can see that there could be hundreds of guide RNA. This is one is C. elegans, the second humans. They have also have a very huge number of guide RNA binding at a particular region. We detect that. So meaning that one region multiple targeting is achievable by our method. Another screenshot, I'll skip that. So again, the score is also correlated to the GC. That's a kind of for some it's correlatable highly, but for some it's not. We see when you increase the mismatches, we see that the number of guide RNA, getting reduced. So when you have genome mismatch, you have a large number of guide RNA. In our method two, we find a very similar kind of uh, similar kind of results that one would expect. And if you see the guide RNA, these are the nucleotide composition, and we allowed some mismatches. So this is a kind of alignment, multiple sequence alignment here. So we see around in the 23, here is 23 nucleotide, that's a guide RNA. So in this guide RNA, there is, although they have a higher score, most of them have a very good match, but there will be some mismatches here and there. But still, this guide RNA could work. Okay, they all have a similar score. Okay, irrespective of a certain number of nucleotide changes, they have a very good score. That's what I want to say. So also apart from the guide RNA score, how well this guide RNA is dispersed, like how well in the genome they are so there is no correlation here. Like the score, if it is like a very highly dispersed when the guide RNA is high, then you'll see the red bar being one to one like this on a diagonal side. But what we see here, there is no correlation to the dispersion. So guide RNA can bind uniquely, but it may not be distributed throughout the genome in a highly deviated manner, like a very highly dispersed. With this, I want to say that this is not necessary, but still we want to say that the guide RNA score that we assigned is almost perfect, but still there is some issues with that. But in terms of dispersion, we still the guide RNA score is still relevant, although the dispersion is not correlating here. Um, uh, so it's like selling up our tool. So our tool is the better performing now with both the C ligands and also humans. Okay, there is no but our method kind of minimize the potential number of off targets. So we try to get the red ones, which is nothing but which are targeting directly to the specific genic regions of interest. So those are like if you select a certain guide RNA, you can also do the multi-targeting in both species based on the selection of the guide RNA. And also when you increase the size range of the window, for example, if you go from 10 kb to 50 kb, the 50 kb range of the genomic region, then the number of guide RNA selectable is very high. So higher the size of the window, higher the possibility that you could find some kind of certain kind of guide RNAs. And some of the references for these tools, you can find it online. I listed out it here. These are like a list of some of the tools that is used to detect guide RNA. So with this, I want to wind up, and I will take any questions that if you have any.
So thank you, Dr. Alaguraj, for excellent presentation. Actually, now the floor is open for questions. Okay, so you can type in the questions in the chat box also. Sir, I have a question actually. Do we have this particular tool available online, or is it downloadable for you?
Yes. Okay. That's a correct question. Actually, this is available in GitHub. What do you call it as? There's an open source software repository.
Okay, okay, okay,
So this is available as a source code. It's a Python-based code.
okay,
It is a platform independent, so you can download it and run it on a system.
okay.
Yeah.
So, okay,
The
is it like
input
is
is like a DNA sequence and the feature of the length of the guide RNA and so on. So.
okay, okay.
Mhm.
So GitHub link we can just search like that, right? Or you have link or something like that. If we can put no, we can like or
Okay.
okay,
Okay.
maybe
Uh,
or
um,
else you can send to me the later. Also, I will put that. No issues.
okay. Okay. I will send the link. Actually, if you can search in my name in the GitHub,
Okay,
you could, or you can just put CRISPR broad, the tool name. You could
okay,
find that. Mm.
okay, okay.
Mm.
So participants can ask questions. Okay, you can either directly ask or you can type in the chat box itself so that the speaker can answer. Okay. So what exactly the medical applications of this CRISPR decast system actually? Can we discuss on something on that? So,
Yeah, medical application. I mean, for the it's mostly most easy to use on single cell systems or cell systems. It's very easy. You can people can deliver the dcas9 and the guide RNA can out of the plasmid and the enzyme, and then there are people can directly. Uh, Transfer these things into the nucleus. It's easy in the cell system. You can put it in a test tube and then you can transfer it. But in plant system it's also easier because you can manipulate the seed and the embryo. But in case of humans it's for diseases it's a little difficult. uh, because of the delivering this will be really be difficult, and targeting to particular cell types is very difficult. Targeting to a particular genomic regions is easy, but delivering this to a particular cell type will be difficult. But people are currently applying it to embryos. Maybe in the future, definitely, this could be coming in a different deliverable format, and then it could make a lot of changes in the diseases. For example, in the US, I mean, the CRISPR system is a patented one. It's a patented, and they have a put it in IPO. Means that means it's in the stock market. So any disease that can be treated with this CRISPR system will be. I mean, people will be paying to the US for that. So they're expecting like in few years time this will be like a very huge success. Like there will be a lot of Mendelian and also non-Mendelian diseases that can be treated. So people believe like that. Mm.
okay, so thank you.
Mm.
So Sibu sir, you have a question there? Sibu sir is there?
Yeah. So so you showed the example of Homo sapiens and C. elegans. So applying your tool into these organisms, but then how about we can, how extent we can use it in plants actually, Arabidopsis, for example.
Okay, that's a good question. Actually, the plant and the C. elegans systems are like very similar. They do not have like animal system like a very large number of repeat sequences. So the humans have a lot of retro transpose on other sequences. So the limitation of this program on applying on any genome is the repeat unit. Okay, so if it is very highly then it's going to be a little difficult. But for plant systems it's really easy. For a plant system like Arabidopsis, it's the program can be like you can manipulate it. You can just give input in the DNA sequence that you have, and then directly manipulate it. It's 5 chromosome. It's going to be easy to run. So But we made it like this, you can input any genome, any plant or any genome, prokaryotic genome too. You can put any genome,
Yeah. Okay.
but the runtime may be going to be little different if it is very highly repetitive genome. Runtime will be very huge. We benchmarked it for Arabidopsis size genome. It may run for 3 hours.
Okay.
So yeah, so yeah.
Okay. So it's already got published. So you already so published this one, or you are still you are releasing it as a software? What is the status of it?
This is under review actually. This is in
Okay.
the second review actually.
Yeah. Yeah.
Yeah,
Okay. Yeah.
yeah. Maybe that's why it's the link is not directly there, but it's in few weeks time maybe it will be up. Yeah.
Okay. Okay.
Yeah.
Very nice presentation, Alaguraj. Thank you.
Thank you. Thank you.
So there's a question from Remya S. Actually, the question is, what makes the GC content an important factor?
Okay.
Yeah.
That's a great question. GC content, what it makes is because uh, when you put the guider in the DCAS9 to deliver it to bind to target to a particular region, the binding is very stable. Okay, when you have a very high GC content. Okay, why if it has a very low GC content with a high 80, what is its problem? This binding may not be stable until this genome editing happens. Okay, there's a time frame for the binding and the editing, and also this, I think it's a very common if you do a PCR. Okay, if you do a PCR for the PCR primer design, people still use that. The complementary sequence to the PCR sequence should be, the primer sequence should be very high GC, right? So that will be very stable when you do the kind of PCR cycling. So so in the same way here, it's a editing, and then there is a polymerase reaction happening. So in this case also you need a stable binding. So you need a that's why people use a GC content more than 50% is very good. 70% is very good. So most of the genomic manipulation, it's a kind of drawback that the nucleotide sequence have a lot of 80. They need a lot of GC. Yeah.
One more question is there regarding from Remy actually. Suppose if the guide RNA do not bind to the target site and bind to the site having same nucleated sequence, then what shall be the consequence?
Okay, yeah, that's a good, important question. There is a consequence. Okay, the guide RNA, if it is not binding to the exact target, and if it goes to other region, it will lead to other diseases, right? Too many other problems. They do not apply humans. That is one of the reason actually. They try because we don't know what is. Okay, for example, people design low GC content guide RNA, and it's not unique to and do not bind to the exact target. If it is low, then it goes and bind to any other region and just modify it. That is very, very dangerous, right? So this technology is still evolving. Still, people are trying to improve it, but only in certain cases the target is unique. In other cases, the target is not unique, and it leads to unwanted consequence. So this is a kind of a two-sided sword. So one person who uses can also get a problem. So it needs to be used carefully with this guide RNA design. That's why people are keep improving methods on the guide RNA design. So there is a consequence, as the person said, there's a huge problem. So if it has a non-target binding possibility, then people will not apply this method at all. Okay, so there is a drawback in that.
Okay. So another interesting question from Dr. Reena Anthony. Sir, in your view, is CRISPR technology ethical?
Ethical. I think this is a debatable question. Like still people are debating.
Sir, please mute your mic. Okay, other participants, please mute your mic. Yes, sir.
So yeah, so this is a kind of debatable question, like where it should be used in plants. Is it okay? But if it is properly used, you can avoid a lot of childhood diseases. For example, there are n number of neurological problems. There is n number of, for example, in one mutation, only one single mutation in tomato is causing the, it's limiting its ripening. Okay, limiting its only single mutation in one gene. So this can be, in this case, it's really ethical because it's not going to affect the, it's going to improve the production. So there will be a food security there. So sustainability will be there. So in that case, for example, most of the plants that we use now is genomically modified. Okay, it's like either by breeding methods. Farmers, they breed with other good varieties. They change the genome. By that method, they change the genome. We are doing it artificially in the lab, but what farmers do is crossing the two species is really changing the genome. So it's on plants, it's really going to be, it's going to be advantageous and ethical. And with humans, it's really debatable whether what are the consequences, what are the, in the future what will happen. It's like taking this COVID vaccine. Okay, it's like is it safe, or and after 20 years what will happen? But still we can take it for some of the diseases. The same way for life threatening diseases, it should be ethical, right? Some life threatening disease, somebody is going to be at the terminal end of the life, then you can use that to, in that case it's ethical. Yeah.
Okay, sir. So another question from Srividya. Sir, is it possible to construct a guide RNA for site-specific RNA editing?
Yes. RNA editing is another method too. You can design the same way for the DNA. You can design the same way, put a RNA complementary sequences. So you can edit the RNA, but the problem with the RNA editing is should be like it's very unstable. Okay, the RNA that you want to edit is going to be very unstable. It's difficult, but people still do that. But in that case, the genome editing is much easier than the RNA editing, right? So you can if you want to activate it, if you want to change the modification, if you want to change the nucleotide, you can do that. But RNA editing is possible, but still it's a kind of a difficult one. People do that RNA editing too.
Okay.
Yeah.
So in line with the question, actually, is it RNA editing more specific? Actually, will it be more specific rather than your DNA editing, DNA binding? I mean,
The specificity is, depends on your guide RNA. Your guide RNA. So you design the same way for RNA, the same thing for the DNA. You design the same. So the complementarity should be the really be the thing. So there will be like a kind of mismatches within the complement sequences. So that's the important thing. And also where you want to edit, whether this sequence is exactly within that region or little bit away from the targeting sequences. So these are the features, but whether it is DNA or RNA may not make any difference. It's only the kind of design, the kind of feature that's going to be matter much. Yeah.
So another question is from Prajita Jain. That is how the genome editing tool CRISPR has been applied to fight against COVID-19.
COVID-19. I don't see something of, uh, but any people try to use on HIV too. They can, just design a complementary sequence, and then, but the problem is just you can, think you have the DNA, human genome is pretty much stable, right? From your birth to the death, it's pretty much stable. It's the same nuclear. But that's unlike, human, the COVID, coronavirus is very unstable, right? Their genome is like mutating forever, right? You have one for the Indian, and actually within India you have multiple mutant here within India. We didn't sequence it, but the person in ICGB, IGIB, they have sequenced certain, and then they put a phylogeny, and you see the sequence is diverse. Okay, so when you make a CRISPR based design for one of them, it will not work on the other. It will be very difficult to design virus based CRISPR guide RNAs and targeting it. It should be stable. If it is a stable virus without a random mutation happening, then possible it is. But the RNA viruses are RNA, and these viruses are really mutating faster. It's expected. So it's difficult to design CRISPR tool for that, right?
Yes. Okay, so another question is from Deepthi Suresh Kumar. The introduction of CRISPR-Cas9 in plant breeding is opening up new approaches for crop improvement. Where do it is mostly employed, most effectively employed? Plant breeding.
I see. I would say, for example, from my recent reading, I see people use it in tobacco plants. They want to, because that's highly funded, because tobacco plant is like a, they want to improve the crop. Yeah, but still, certain labs also use it on rice. Um, as I say, tomato, but most of them are like benchmark. Like I think Cebu should be, commenting on that. They are mostly on the Arabidopsis. It's because that's a model system. But for the crop plants, I would say from what I know, I think it's mostly on the rice, tomato, and things like that. Cebu, do you want to comment?
Sir?
Yeah, actually, it's everywhere. All the major crops, they are adopting CRISPR now. I mean, that is the benchmark now. I mean, nobody is staying behind, because everybody knows that this is the future. The only thing is, another advantage is that CRISPR is kind of allowed in United States, because the amount of foreign DNA what we are introducing is small enough to not to recognize this as a genetically modified organism. So this is the one thing, one advantage CRISPR has actually. But in Europe and other places, still people are considering this one as a genetically modified organism. So, but still, I mean, even irrespective of the place, people are still identifying the potential of the technology, and they are taking up this one as the major technology for gene editing. So, I mean, no one is staying behind because of all this controversy. So everybody is still working on that. That's what I think. Yeah.
Yeah, I think most of the plants, they have a preliminary judgment. Currently the tool is available, but the question is what trait you want to change.
Yeah.
Okay, which changes will lead to good crop improvement. That's the tool is there with you, but the thing is where you want to, which region in the genome you want to apply. That's going to be the difficult
Yeah,
thing.
it's true. Which trait to choose, which trait to choose. Yeah, that is the crucial decision, I think.
Yeah, crucial. So people try to use a kind of library, like the hundreds of regions. They try a combination of regions. They want to change it. They try change it, and then see what is the crop, what is happening to the crop. So they kind of a CRISPR library they are creating, and then try to use that. So I think it's still going. I feel in the plant sector, it's going good.
Yeah.
Okay, sir. So another question has been same line asked by Annie, Miss Annie. So is CRISPR technology is a powerful tool for genome editing plant actually? So I think that answered that question also. Let us move on to the next question. So actually, other question is from Stacy Teresa Tommy. That is, what are the other scientific uses with the CRISPR beyond genome editing?
Yeah, this is something. See, I didn't put some of the uh, apart from genome editing and also the epigenome editing, there is like n number of stuff. For example, here I didn't cover some of the. Uh, um, for example, if you want to target a molecule, forget about editing. Okay, forget about editing. If you want to transport a particular chemical to a nucleotide position, for example, my DNA is like this. Okay, my DNA is wound around the nucleotide, the nucleus. Okay, I want to see where my nucleotide, the particular gene is in the nucleus. Okay, there is no modification here. I want to see, just want to see in the cell where is my gene. Okay, whether it is in the center of the nucleus or in the periphery, people want to image them. Okay, this can be imaged. So what they do is they have the CRISPR based, without the Cas9 activity, they add a chemical group that can go and paint that region. Okay, so the guide RNA is is there. My gene is my, I know the gene where, but I want to see where it is. So they add a paint to that, so it go and paint that region. So you get, and then you can see it in the microscope. Okay, it's a kind of a tagging. So you can see in the microscope whether my gene is. For example, one easiest the question is, I am going to introduce a tomato gene to a potato. Okay, I introduced a gene which is not there in potato, but it is in tomato. So I cut it and put it in potato. Okay, I put that gene in potato, but how do I see whether that gene is there or not? You believe that I already inserted, but I want to see whether it is already there. So in that case, what they do, they design a guide RNA for that tomato gene, and then they have a paint for that, and then they put it there, and the CRISPR will not edit the genome but go and paint that gene, and then you can see that, yeah, your gene from tomato is transferred to potato. You confirm it, right? So this kind of painting experiments, that's what you call it as imaging. And also, if you want, for example, my protein, enzymes are there in the genome. Okay, some enzymes are there in the cell. They may not bind to the genome. Okay, because of some kind of mutation or some kind of there are other possibility that the enzyme cannot be functional. So the enzyme may not be targeted to that genome. So in that case also, what they do, they want to target this protein, so they will tag this protein to the guide RNA, and then the protein will go and directly bind them, because the complementary guide RNA, so it will transport. So one is imaging and the second one is transporting. Okay, it's not going to edit the genome, but it's going to transport that protein. Okay, so they will tag it, and then that will tag. So anything that can be transported to the genome can be done using CRISPR, not only editing. Okay, so these are some of the stuff. I mean, as I said, genome editing is one, but the epigenome editing is also one. That you can remove unwanted chemical groups that is added to the genome. Just you can add a particular chemical group to the genome to see what are the changes. So there are, I think that in the future there are other applications. There should be a more number of applications. Currently, these are the main applications.
Okay, okay, sir. So another question is there from Sangeeta actually. Sir, is there any way to ensure that CRISPR-Cas9 binds accurately,
Accurately.
specifically? Yeah, yeah.
Okay. So one is directly finding the if you take the guide and then you can blast it on the genome. Okay, just see, blast on the genome, and then you see whether it can bind certain certain region, certain sometimes your guide RNA may be designed, but it may not have worked in the cell. So possible that it may not have worked in the cell. So in that case, there is a benchmark that certain amount of guide RNA should be introduced for a binding or a particular targeting. So if it exceeds a certain that amount of guide RNA needed, the people have benchmarked actually for a particular species, particular kind of activity. They have benchmarked that they need only this amount of guide RNA. But for example, one guide RNA. Sometimes it requires two guide RNA. So things like that. So the one way is just to make sure that you check the sequence, that the sequence is not duplicated at all. The guide RNA sequence is unique. That way, one is true. The second way is the amount of guide RNA that one could put, and the number of guide RNA one could put for the molecule to make the changes to make that targeting. So for if it is targeted or not, you can check the specificity by means of increasing the guide RNA to the effect. For example, uh, if you take activation of gene expression, you need to put a kind of ROC curve, or what do you call it, as a saturation curve. You put certain level and then it should increase. When you increase the guide RNA, it should increase the level of expression. So that way one could see that it's really going correct, something like that. So the sequence complementarity, and also the amount of guide RNA and the number of guide RNA one needs. Those are the benchmarking now. People use that as a benchmark now.
Okay, sir. There is a final question from an interesting question from Triveni AJ. That is, can this multi-drug resistance of bacteria can be reversed using this method?
Multi-drug resistance.
Yes, yeah.
Can be reversed.
Yeah, that's right.
I'm not sure, but the resistance comes from what? Resistance comes from
maybe we can is it easily able to bind out the particular gated RNA to the bacteria so that you can do something on antibiotic resistance? That may be the question. Right?
Antibiotic resistance comes from is it coming from a sequence? Is it coming from a mutation? I'm not sure. I don't know. I'm not very sure about how this multi-drug resistance in bacteria operates. If I understand a little bit, I could actually comment it, but I'm not sure about the multi-drug resistance. How the bacteria achieve that multi-drug resistance. Is it like a protein level changes, or is it based on the nucleotide level changes? I'm not very sure about that.
Okay.
Sir, can you speak, sir?
Yes, Triveni, you can comment on that.
Thank you, sir. Thank you so much for the opportunity, and a very good evening, sir. Sir, actually, many of the multi-drug resistance bacteria, they will cause severe infections with biofilm formation. So even in biofilm formation, we will find the DNA. eDNA, secret DNA.
Then it should be a case, right? If you have a drug resistant bacteria that has a unique sequences.
Sir,
if it have a unique
my
sequences.
question, yes, sir, exactly. And these certain bacteria, they will evolve as multi-drug resistance, and one of the virulence factor is biofilm.
Okay.
So can we, by using this method, you said it's like either editing factor, can we edit this virulence factors? Sir, you need to give more clarity on that, sir, whether it is possible or not.
Actually, you see, in this case, you want to remove the bacteria, right?
Remove the biofilm, sir. Remove the
Biofilm.
biofilm.
Yeah, I mean, by that means you want to remove the bacteria from the system, right? Or
Like, sir, uh, sir, for example, many of the bacteria, they will produce the biofilm in the stress condition.
Mhm.
Stress condition, for example, if a person is taking antibiotics, so to trigger that, they will form biofilm so that the antibiotic should not work.
Mhm.
Okay,
Okay.
in such cases, so this will evolve as a strong biofilm forming bacteria.
Mhm.
So is there any methodology so that we can reverse this by using this? Because once it will form biofilms, it is very hard for the antibiotics to penetrate. Can we reverse this, the one which has already become multi-drug resistant biofilm forming, without biofilm forming a normal bacteria?
I think it should be, you need to figure out which, what kind of pathways are involved in that biofilm formation. And you can actually delete out, or you can cut those genes responsible. If it is like the whole pathway number of genes, if they are in consecutively arranged, you can uh, exclude those genes from the bacteria. It should be
Is
easier.
that possible, sir? Possible,
Yeah, it's
because
possible.
for
Because
biofilm formation,
yeah,
for
for
example, ICAD genes are responsible. Now, can we
Mhm.
edit this that part?
Yeah, if the gene is very large in length, or if it involves, you need to figure out if it is one gene or if it is multiple set of genes. Usually it should be a pathway of genes from which source this metabolite to this metabolite, and then finally biofilm formation. So
Yes, sir.
there should be a set of genes. In that case, you may need to have a very complex strategy, like if it is 3 kb or more than 10 kb regions you want to wait out, then you need to design guide RNA such that it piece out into, piece out this into smaller ones without, when you cut into two piece, when you cut a smaller piece out of it, it can reverse and bind. So you have to see, if you are cutting multiple pieces, you need to see that the process is a little complex, that you're going to join the genome at the end, but you're removing the rest of the smaller pieces. So the rest of the smaller pieces should not bind or connect in any way. So it should be possible, but it's really going to be using multiple guide RNAs, okay, not one guide RNA, one Cas9 enzyme. It will be like sending a pool of guide RNA on each region, like covering the whole gene and covering multiple genes. So removing one mutation is different. Removing the whole gene is, a totally different. But it's totally possible with the strategy. It should be little different. Uh,
Okay.
uh, yeah, it's like
Thank you.
one end of the hook and the another end of the hook. You need to design like that, and you need to send two equal amount of guide RNA Cas9 system, so that in all cells, both sides, it's cut down. So people use like that strategy. Yeah.
Thank you, sir. Thank you so much for the wonderful clarification.
Thank you.
Thank you.
So I think that's about all the questions actually. If anybody want to ask any questions, they can ask directly, or you can type in the chat box. Thank you, Dr. Alaguraj, for your excellent presentation as well as your interaction. Actually, I know it's very difficult for you since it's a very early morning Saturday in there actually in US. So Shibu sir, you have anything to comment on that?
No, nothing, Alaguraj. So he's always like that. Whenever we ask for this kind of things, he's always ready to participate. I mean, irrespective of the time or everything. Once I was even asking him to join for something much earlier time than this, and he joined for that one. So he's like that. Now he's always very generous. So thank you, Alaguraj. It was, I know you mean, as I suggested, you prepared such a very simple presentation which can be accessible for almost everyone. Thank you, Alaguraj. Thank you so much for such a nice presentation.
Thank you, Subbu. Thank you,
sir, for
everyone,
accepting
both faculty and students of the.
Yeah.
Thank you, sir, for accepting our invitation and coming for this presentation. And
Thank you, ma'am. Thank you so much. It's
sure
pleasure.
that all the students would have actually understood the whole system without, and we don't have to teach it again anymore for them. So that is that simple for them. Thank you so much, sir.
Thank you.
So by that, we come to the end of our today's session. So I invite Miss Steffi Sosa Thomas, MSc for Technology students at MacFirst, to post the vote of thanks.
Sir, am I audible?
Yes.
Good evening, everyone. As we come to the end of this webinar, on behalf of MacFirst College Tiruvalla, it gives me immense pleasure to deliver the vote of thanks. First of all, my very special thanks to Dr. Alaguraj Veluchamy, who gave a very informative class on the topic Computational Approaches for Epigenome Editing by CRISPR-Cas9 System. Next, I extend my sincere thanks to our principal, Father Dr. Cherian J. Kotel, and Dr. Jenny Jacob, Head of Department of Bioscience, for providing such a wonderful opportunity. A special thanks to Dr. Sibu Simon, the coordinator of this webinar, and program committee members for taking the effort to put this event together. I would also like to express my heartfelt gratitude to our Chief Administrator, Professor Verghese Abraham Sir, our Research Director, Dr. Jyotis Kumar Sir, and Dean Ms. Veena Cherian Ma'am. Next, I would like to thank all the participants who made this webinar a great success. I also express my sincere gratitude to all the faculty members and my dear friends for your attention and cooperation. Once again, thank you all.
Thank you, Steffi, for your vote of thanks. Thank you, sir, actually. And all the participants, please fill in the feedback form, which is given in the chat box link. Thank you all. Thank you. And date, doesn't matter, date is also given there. So you can fill in the feedback form, actually. So the certificates will distribute after that.
Okay, all then. Alaguraj, thank you then. Bye. See you. Bye.
Thank you, sir.
Thank you.